I am Rinjuna Reenas, Assistant Professor in the Department of Pharmaceutical Chemistry, Yenepoya Pharmacy College and Research Center. Today, we will be discussing on Compexometric titration. These are the contents which we will discuss, which will include introduction, based on ligands, PM indicators, masking and demasking agents. Specific learning outcomes. Students should be able to explain the principle of complexometric titration at the end of the session, and students should be able to explain what are ligands and its types, and should be able to understand the difference between masking and demasking agents. Complexometric titration. Complexometric titration is a type of titration which is based on the complex formation between analyte and titrant. It is also known as chelatometric titration. It is mainly used for the determination of metal ions in the solution. So, here the analyte solution will be metal ions and the titrant will be is also known as ligands. When a metal ion combines with a molecule which can donate an electron, the resulting compound is termed as complex. The molecule bound to the metal ion is called ligand. In complexometric titration, metal ion accepts electron and ligand donates it. Thus, in a ligand molecule, there is a presence of at least one lone pair of electrons through which a coordinate linkage with the metal ion take place. Ligands are the molecules or ions which are coordinated to the metal ion. For example, potassium ferrocyanide, there are 6 cyanide group. These are the ligands which are attached to our ferrous ions. Ligands are also known as Lewis bases because they can donate electrons to our metal ions. So, metal ions are also known as what? Lewis acids. This is the structure of EDTA, which is known as ethylene diamine tetraacetic acid. EDTA is one of the most commonly used ligands in our complexometric titration. Classification of ligands. Based on the number of points of attachment to the metal ion or number of electron donating groups present in the ligands are classified into monodentate ligand, bidentate ligand and multidentate ligand. Monodentate ligand. Monodentate ligands are those compounds which are having one electron donating group in them or which can donate a lone pair of electron to our metals. For example, ammonia. In ammonia, only one nitrogen atom is present so that it can donate one lone pair of electrons to our metal atom. Bidentate ligand. Bidentate ligands are those compounds which can donate 2 electron groups or which are having 2 donor groups in them. For example, ethylene diamine. Ethylene diamine is having 2 nitrogen, which means it can donate 2 lone pair of electrons to our metal atom. Next is multidentate ligand. These ligands contain more than 2 coordinating atoms per molecule. The ethylene diamine tetraacetic acid has 2 donor nitrogen atoms and 4 donor COO in the molecule, which is thus by it is hexadentate. That means it is having 6 points of attachment to our metal atom. Next is metal ion indicators. Usually we will be using indicator in order to dictate the end point in a titration. So, the PM indicator or metal ion indicators are those compounds which we are using in complexometric titration in order to determine the end point of the titration. The most commonly used PM indicators are eriochrome black tea, xylenol orange and muroxide. For example, consider titration of sample containing calcium, magnesium or any other metal atoms in a conical flask. To this flask, if we are adding our indicator, for example, eriochrome black tea means our indicator initially will form a bond with our metal ion. The color of that solution will be wine red in color. But once we start our titration with EDTA, this EDTA will replace our indicator from the metal indicator complex. So that indicator will be left alone, and this left alone indicator will have a different color. That color will be blue. The color of the free indicator will be blue and the new complex formed between the EDTA and metal will be colorless. This is how a color change or endpoint detection in a complexometric titration is taking place. For example, you can see here the calcium and magnesium is taken in the conical flask. To this, if you are adding an indicator, means the indicator will initially form a bond with our calcium magnesium, or a complex will be formed between indicator and metal ion whose color will be wine red. Then once we start doing the titration, the ETTA will replace the indicator from the calcium indicator complex. A new complex between EDTA and metal will be formed and our indicator will be left alone. And this free indicator is having another color which is blue. This is how the end point is determined using PM indicators. This Aerochrome black tea is used to determine the concentration of calcium, magnesium, zinc and cadmium, and the free indicator is blue in color, but upon the complexation with the metal ion, a pink color is produced. Masking agents. When it is required to assay selectively one or more ions in a mixture of cations and to eliminate the effects of possible impurities which would add into the titer, masking agents are used. Masking agents mask the presence of particular cation and allow the titrations of the specific cations with the complex forming agent. That means in a conical flask, if you are given a mixture of metals, cadmium, lead, zinc, etc., calcium, etc., but if you want to selectively titrate with any one of the metal ion, you can use masking agent. This masking agents will inhibit the interference of other remaining metals during your titration. Thereby, we can selectively titrate with the specific metal ion. For example, here in when the cuprous ion react with iodine, if iron is present in them, means the iron will interfere in this reaction. So we can avoid this by adding some fluoride ion. So here the fluoride ion will act as masking agent and a complex between fluoride and ferrous ion will be formed. Thereby, if you are again in this reaction, that is reaction between cuprous ion and the iodine, this won't interfere. Next one is demasking agent. Demasking is the process by which masked substance regains its ability to enter into a particular reaction. That means if you are already masked a particular ion and if you want that similar metal ion to enter again into the reaction, you can use a demasking agent. For example, formaldehyde and zinc. Zinc is already masked, and if you are adding formaldehyde, means formaldehyde will free the zinc from that complex, thereby zinc will be free. Now if you are again titrating it with ETTA, means zinc will enter in the reaction. Thank you.